I'm Alejandro Martinez, and I'm going to be talking to you about primary structure. I'm going to be talking to you about primary, secondary, tertiary, and quaternary formation in a protein. So here is a brief overview of what I'm going to be going over in this video. I'm going to be talking to you about proteins, amino acids, bonds. I'm also going to be talking to you, of course, about the primary structure, secondary, tertiary, and quaternary. And I'm going to be explaining those throughout this video, so stick along. I'm going to be giving to you a short description on proteins, and we know that protein is a macromolecule. It's one of the four macromolecules of life. It's up there with the polysaccharides, the lipids, and the nucleic acids. And we know that protein is a polymer, and a polymer is just a large molecule. And the large molecule consists of monomers, which are smaller molecules that come together and make a polymer. So, we have protein, a macromolecule, and it's a polypeptide. And I'll show you what that means in this little illustration. So the AA is an amino acid. Those are the monomers that make up the protein. And you have these little covalent bonds, and those covalent bonds are peptide bonds. And when you have multiple amino acids bonded together by peptide bonds, you form a polypeptide. And a protein is composed of one polypeptide or more. So we can use them interchangeably if we use it correctly. So here we have a picture of a amino acid, and we can see on the left side of the amino acid where we have a nitrogen and two hydrogen atoms. That part of the amino acid is what we call the amino acid group, and then the H on top of the carbon in the middle in the red box, that's the hydrogen of the amino acid. And then towards the right of that hydrogen, where we have two oxygen atoms and a carbon and a hydrogen atom, that's what we call the carboxyl group of the amino acid. And then lastly, we have the R group, and the R group is going to be different for every amino acid, and we roughly have about 20 amino acids, so that's going to be different. And like we know before, there are three types of amino acid. There is polar, nonpolar, and polar charged, and all of this is going to be important when we talk about the structure in the protein. So the first structure I'm going to be talking to you about is primary structure, and what primary structure signifies is the order of the amino acids in a polypeptide. So here I color coded three amino acids. We have the yellow, the red, and the blue. Now, when we describe the primary structure of a protein, we're going to use the first three letters of that amino acid. So it would be the first three letters of the yellow amino acid. Then we have our dash, the first three letters of the red amino acid, and then the first three letters of the blue amino acid, and this would go on because that's how we describe the primary structure of a protein. So the next type of structure we have is called secondary structure, and this is the way polypeptides fold onto itself, and it's between the amino and carboxyl groups. It's when the polypeptides fold onto itself, we get two type of motifs. We have the alpha helix and a beta pleated sheet, and I'll describe more of that later. So when a polypeptide folds onto itself, we either get alpha helix or beta sheet. And with alpha helix, we have hydrogen bonds that run up and down the alpha helix, stabilizing the protein. And we have beta sheet, which has two forms, parallel and anti-parallel. And the difference is that with parallel, we have the carboxyl groups and the hydrogen groups that are lined up. And with non-parallel, it's they're not lined up, and that's the difference between parallel and anti-parallel. The next type of structure we have is called tertiary structure, and that is the three-dimensional shaping that comes when a polypeptide folds themselves one more time, and we have a sort of three-dimensional a polypeptide, and there are many factors that contribute to that shaping and the appearance, and one of those being is hydrogen bonds. We also have ionic bonds, hydrophobic bonds, and the disulfide bonds, and I'm going to explain these later in this video, so stick along. So, like I mentioned previously, hydrogen bonds are important because they affect the tertiary structure of a polypeptide. And these hydrogen bonds usually occur in the side chains in the amino acid, and they're usually between two alcohols, an alcohol or an acid, or an alcohol and an amine or an amide. And these are, like I said, crucial to understanding tertiary structure because they affect the polypeptide shape. Next structure that affects tertiary structure is called ionic bonds. And what happens is the neutralization of an acid and an amine create these salt bridges, and these salt bridges have the effect of straightening an alpha helix. And what happens is ionic because you have positive and negative groups that when folded come to close contact with each other. And you all know the saying, how positive and negative attract, whereas positive and positive repel, and the same thing with negative and negative. And these have the effect of tertiary structure, is what we take into consideration when determining that tertiary structure of that protein. So the hydrophobic effect, influences the tertiary structure of a polypeptide, and we first have to revisit the idea of polar and non-polar. Well, remember that polar, think of it as polar loving water and non-polar loathing water. So, we have let's say we're in a watery region of a folded up protein, and we have the protein, and, we have the polar is going to be on the outside, while the non-polar is going to be on the inside, and that's going to is going to greatly affect the structure of that polypeptide. Another factor that affects tertiary structure are called disulfide bonds. And what you have is a cysteine and a thiol group. And in the thiol group, you have a sulfur atom. And that sulfur atom wants to oxidize. And when it does that, it forms strong covalent bonds. And that affects the structure. So the last type of structure is called quaternary structure, and that's between several polypeptides. And all the different factors that encompass tertiary structure affect quaternary structure. So you're going to see the same thing. You're going to see, cysteine. You're going to see, hydrogen bonds, hydrophobic effect. You're going to see all of that in quaternary structure.